Okay, everyone, here we are back for the fourth and final part of the video lecture for Chapter 5 on large biological molecules. At the end of the last chapter, the end of the last video, we had finished talking about protein structure and function, the way proteins can unfold or denature and renature and all that. The last category of biological molecules excuse me, that we're going to talk about in this chapter is the nucleic acids. So nucleic acids basically are DNA and RNA. Now, most people have heard of DNA, probably also RNA. These are the molecules that are involved in heredity, particularly DNA. DNA is the hereditary molecule. It's the molecule that allows for passage of information from one generation to the next. It's what we get from our parents. Half of your DNA came from your mother, the other half came from your father. And then your body is built based on the information in that DNA. Okay, so with nucleic acids, we're still talking about that monomer-polymer plan for molecule construction that we talked about with carbohydrates, that we then talked about with proteins. Remember, we got away from that with the fats, the lipids, because they're not built that way. But carbohydrates, proteins, and now nucleic acids definitely are built on this monomer-polymer plan. Okay, so the amino acid sequence of a polypeptide that we talked about in the last video lecture is programmed by a unit of inheritance called a gene, okay? Sorry, that's a G. The gene. The gene, for now, just think of it as a stretch or a segment of DNA that codes for a protein. Our genes contain information for how to build proteins. In particular, what your genes do is they tell your cells in what order to link amino acids together to build a polypeptide, which then folds up into a protein. So the genetic information in your DNA literally is the building code for how to link amino acids together to build proteins. That tells you how important proteins are. Your DNA, which we tend to think of as, you know, this molecule that instructs how to build our bodies and how our bodies work, does one thing. And that one thing is that it builds protein. It provides the instructions for building proteins. Your proteins then go out in your body and do all that work. That tells you how important proteins are if DNA is the instructions for building protein. All right, so DNA is short for deoxyribonucleic acid. Deoxyribonucleic acid. RNA is short for ribonucleic acid, all right? And again, both of these are polymers made up of certain monomers that we'll get to in a little bit. Think of DNA as a storage molecule. Okay, it stores information. It's a code, essentially. It also provides directions for its own replication. It's a one-stop shop. It provides information for how to build proteins, but also it includes the information that the DNA needs to copy itself. And we'll talk about how DNA copies itself in a lot of detail in Chapter 16. But DNA also provides instructions for how to build RNA, okay? In particular, what we call messenger RNA, or mRNA for short. Think of messenger RNA as sort of like a copy or a photocopy of the DNA. So we have a process whereby information flows in our cells from DNA to messenger RNA, and then from messenger RNA to protein. That process of information flow from DNA to messenger RNA and then messenger RNA to protein is called gene expression. All right, gene expression. This is how the information in our DNA is ultimately used to build protein, all right? So we'll talk about that also in a lot more detail in the very last chapter for the course, Chapter 17. All right, so let's go back to nucleic acid structure. All right, well, we have, again, monomers linked together to build polymers, but the monomers are not monosaccharides. That was carbohydrates. they're not amino acids, that was proteins. The monomers here are called nucleotides. So these are the monomers that are linked to build DNA and RNA. The polymer is called a polynucleotide, okay? That's our polymer in this case. And DNA is a polynucleotide, RNA is a polynucleotide. Okay, so now we need to talk about the structure, first of all, of these monomer building blocks, okay, the nucleotides as we call them. All right, so let's take a look. All right, here we have some information about structure of nucleotides, but also of the polymers. Okay, look at this panel B here first. This is a single nucleotide monomer. That's one building block. Every nucleotide, and there are 5 of them in nucleic acids, not 20 like in proteins, but 5, every one of them is built the same way. There's part of the molecule called a nitrogenous base. This just means it's a part of the molecule that contains nitrogen. There are 5 different ones: cytosine, thymine, uracil, adenine, and guanine. Okay, now in DNA we only have 4 of these bases. In DNA we have cytosine, thymine, adenine, guanine. So C, T, A, G. In RNA we also have 4, but they are a little bit different. In RNA they are cytosine, uracil, adenine, and guanine. We don't have thymine in RNA, and we don't have uracil in DNA. All right, so every nucleotide has one of those bases. If it's DNA, the bases are A, T, C, G. If it's RNA, they are A, U, C, and G. All right, that base is then connected to a 5-carbon sugar, a pentose sugar. In DNA, the sugar is deoxyribose. In RNA, it's ribose. This is why they're named different molecules: deoxyribonucleic acid or ribonucleic acid. Then that sugar is connected to a phosphate group. Phosphate groups are one of those functional groups we talked about earlier, okay, in the last chapter. So that's the structure of a nucleotide: one of 4 or 5 bases, a sugar, and a phosphate group. Okay, how are these linked together? Well, there's going to be a bond between the phosphate group of one nucleotide and the sugar of the next one. So look over here. Here we have a single nucleotide: base, sugar, phosphate. Here we have a second nucleotide: base, sugar, phosphate. There's a bond right there between them. That bond is between the phosphate of this nucleotide and the sugar of that one. That bond is called a phosphodiester bond. Phosphodiester bond. All right. And that's it right there. You see we have a second one here and a third one. It's always between the phosphate of one nucleotide and the sugar of the next. And that those phosphodiester bonds create a backbone called the sugar phosphate backbone. Sugar phosphate backbone right there. Then we have the various bases coming off, just like the R groups of a polypeptide do, to form what's called a side chain. And the side chain, in this case, is the genetic code, the sequence of bases. So maybe that's a guanine, maybe this is cytosine, thymine, for example. That's the genetic code. You read that from one end of a gene to the other, and you have read off the genetic code that then determines the amino acid sequence of our proteins. Okay, so that's essentially the basic structure of any nucleic acid. Now, in DNA, what you'll have is a second chain, okay, a second strand of DNA right here. So you'll have a sugar phosphate backbone with its bases coming off, and the bases will pair up with each other, adjacent to each other, and bond together. And DNA, therefore, is double-stranded as a result of that. We'll see more about that here in a little bit. RNA is typically single-stranded. It can be double-stranded in some cases, as we'll see, but for now, think of RNA as single-stranded. All right. So, talking about the structure here of this molecule, DNA in particular. Again, DNA is double-stranded, RNA is single. Here on the right, in this video I'm getting ready to play, you see a single-stranded RNA molecule. So in pink, you see the sugar phosphate backbone with the various bases coming off to the side. And notice that in this case we have U's. So uracil there, uracil, uracil, and uracil. You don't have thymine or T in RNA. Okay, that's how you know that's RNA. Now, that's single-stranded. But in DNA, we would have another strand over here, and the bases would bond together. And when you do that, they coil up into a spiral structure, okay, what's called the double helix. And that's how this DNA molecule is being represented here. So we have one sugar phosphate backbone there, shown in blue. We have a second one here, shown in blue, and the bases that come off as side chains hydrogen bond to each other in the center. So think of this, if you think of it as a ladder, right? The ladder, the two vertical supports would be the two sugar phosphate backbones. The rungs that you would step on to climb the ladder are the bases paired together in the middle, bonded to each other in the middle by hydrogen bonds. Now take that ladder, both ends, and rotate them in opposite directions, and it coils up into a spiral staircase. That's what you see here on the left for the double helix, with the two handrails of the staircase being made up by the two sugar phosphate backbones, and the steps that you would step on being made up by the bases bonded together in between them. All right,
DNA and RNA are nucleic acids, polymers made of subunits called nucleotides. One difference between DNA and RNA is the type of sugar their nucleotides contain. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. Ribose has one more oxygen atom than deoxyribose. DNA and RNA are each composed of 4 different nucleotides, which differ in their nitrogenous bases. 3 of the 4 bases are the same in DNA and RNA: adenine, guanine, and cytosine. The 4th base in DNA is thymine. In RNA, it is uracil. The nitrogenous bases guanine and adenine each have 2 linked rings of atoms. They are called purines. Cytosine, thymine, and uracil each have a single ring, and these 3 bases are called pyrimidines. For convenience, the carbon atoms in a nucleotide sugars are numbered, beginning with the carbon atom bonded to the nitrogenous base, moving around the ring, and up to the carbon that is bonded to the phosphate group. The 1' carbon is bonded to the nitrogenous base, the 3' carbon to the next nucleotide, and the 5' carbon to the phosphate group. DNA and RNA are polynucleotides, long chains of nucleotides. Polynucleotides are always assembled in the 5' to 3' direction. A covalent bond forms between the carbon at the 3' position of a nucleotide and the phosphate group at the 5' position of the next nucleotide. RNA usually consists of a single polynucleotide chain. DNA, on the other hand, consists of 2 polynucleotide chains. The 2 DNA chains, or strands, are oriented in opposite directions and held together by hydrogen bonds between the nitrogenous bases on opposite strands. Because of their sizes, shapes, and arrangement of polar groups, the DNA bases form complementary pairs. Adenine pairs with thymine, and cytosine pairs with guanine. The 2 polynucleotides in DNA wind around each other to form the familiar double helix. The bases can be in any order, like the letters of the alphabet. However, the base sequence is significant. Sequences of bases called genes encode the instructions for the structure and function of an organism.
Okay, so one thing I want to talk about real quick is that whole 5', 3' thing you just heard about in the video. The information in DNA, the information that our cells use to build proteins, flows in one direction, okay? And so in the same way that if you're reading a book that's written in English, you read from the left-hand side of the page to the right-hand side of the page. The sentence makes sense that way, but try and read it backwards from the right side to the left, and it makes no sense. So there's directionality in the information flow of the language. There's also directionality in the information flow of DNA language, and that's what this 5' 3' has to do with. So remember, the numbers 5 and 3 refer to the sugars in, or I'm sorry, the carbons in the sugars, okay? So one side of the sugar has a 3' carbon on the bottom side. The top side of the sugar has a 5' carbon. So the bottom side is called the 3' side of the sugar. The top side is the 5'. It's no different than saying, you know, your right hand is on the right side of your body and your left hand is on the left side of your body. It's no different than that. All right, so if I draw a double-stranded DNA molecule here, so these are sugar phosphate backbones, and let's say I have the bases pairing up in the middle, so A here always pairs with T there, C here always pairs with G there, and let's say we have a G here paired with C, and we have a T here paired with A. If this is the 5' end on the left strand, that makes this the 3' end. That means that we have a 3' end on the right-hand side and 5' there. Notice how the two strands are running in opposite directions from each other. That is a situation referred to as anti-parallel. The two strands of DNA run in opposite directions, or the information in them runs in opposite directions, all right? It'd be like having two sentences or two lines of written language in English. One, the way we would normally think of, oriented from left to right. The other one flipped around backwards, oriented from right to left, lying parallel to each other. The information in them would flow in different directions. That's anti-parallel. They're parallel, but the anti refers to the fact that the information in them runs in opposite directions. In addition, notice how I've drawn this here, with a cytosine always pairing with a guanine, and a thymine always with an adenine. That is a situation in DNA referred to as complementarity. Okay, DNA and RNA both exhibit complementarity when they are double-stranded. That's a really important part of the coding mechanism of DNA, in other words, in terms of how it stores information. And you see that here, always a C with a G and an A with a T, for example. That means that if you have these strands separated and you wanted to build a new strand onto this one, if you had a T here, an A would have to go there, a C here, a G would have to go there. That's the basis of how DNA copies itself, which we're going to get into next. All right, this last figure from this chapter is showing you essentially what happens during DNA replication. So what we have here in the bluer shades is called the parent strand of DNA, the original strand. Notice we have a 5' end of one strand there. We have the 3' end of the other strand here. That's the anti-parallel. The base complementarity is the C with G and the A with T pairing up in the middle, as you see, right? Now, what happens during DNA replication, which has to happen before a cell divides so that the two cells that result both get a copy of the DNA, is that there's an enzyme that will come along and it will separate the two strands from each other. It sort of snips those hydrogen bonds and separates the two strands. There's another enzyme that then reads these separated bases and says, okay, what nucleotides floating around inside the nucleus of the cell do I need to add here? If it reads a G base, it adds a C. If it reads a C base, it adds a G. An A base, it adds a T, and so on down the line. So it builds, you see in orange here, a copy of that DNA strand. That new strand is called the daughter strand, again shown in orange in this case. If you read this DNA sequence and compare it to this DNA sequence, you'll see they're identical. So we have a coding mechanism here through this base pair complementarity, A with T, C with G, and that's the basis for how DNA copies itself. But again, we'll see a lot more of the details of that in chapter 16, okay, one of our last chapters for the semester. All right, so this wraps up the video lectures for chapter 5 on structure and function of biological molecules.